Good morning everyone. So today topic is bacteriophages, its structure. So in this topic we will learn what is introduction to bacteriophages and its structures. So topic name is bacteriophages. Bacteriophages. So bacteriophage means the virus which attack bacteria or kill bacteria is called bacteriophages. The virus which attack bacteria or kill bacteria is called bacteriophages. Bacteriophages are simply bacteria eater virus, or also we can define as bacteria eater virus. The bacteriophage term was coined by Felix D. Herilly. Felix D. Herilly. The discovery of bacteriophage. The discovery of bacteriophage made by two individual scientists. The first attempt was given by Frederick Twart in 1915, and one more scientist, Felix D. Herilly, in 1917. These two scientists individually they discovered bacteriophages. Next, the shape of the bacteriophage. Shape. Generally, bacteriophage has tadpole shape, tadpole-like shape. And coming to size of the bacteriophage 65 into 95 nanometer in diameter. Now coming to molecular weight of the bacteriophage, molecular weight 1.2 into 10 to the power of 8 Daltons. So it is a molecular weight of bacteriophage. Generally, bacteriophage are any virus, they are obligate intracellular parasite. Intracellular parasites. It means that the virus which multiply or which replicate only in living host. The virus which replicate or multiply only in living host organism. Here the living host organism is bacteria. Now coming to structure of bacteriophages. So this is all about the introduction of bacteriophage. Now we will discuss only structure of bacteriophage. Only structure of bacteriophage. Right. Structure of bacteriophage. Usually virus or bacteriophages, any virus or bacteriophages, usually they are nucleoproteinase structures. They are nucleoproteinous structure mean they contain nucleic acid and protein structure. For example, if you take any virus, they have only two compartments or two parts. One is nucleic acid, and this nucleic acid is surrounded by a protein. So the structure which is made only by nucleic acid and protein, this type of structure is called nucleoproteinous structure, and this is the structure of virus, or we can say any other virus or bacteriophages. And these nucleoproteinous structures generally made up of with two components. One is nucleic acid. Here nucleic acid is act as a genetic material. Nucleic acid act as a genetic material. And second component is protein. Protein are also called capsid. Protein or capsid. Here the nucleic acid or genetic material is the only material which is infectious. It causes infectious. So hence the infectious material here, infectious material is only genetic material, nucleic acid. And generally in mostly bacteriophage has genetic material double-stranded DNA. Double-stranded DNA. Now coming to protein or capsid, these protein or capsids are generally form four parts. Four parts. Like, see here, look at the diagram. In that first four parts, first part is head, second part is collar, third part is tail, and coming to last part is end plate, are also called base plate. Here, so this is the head part, and this is what collar part, and this is the tail part. Here, see, this is the head part, this is the collar part, and this one tail part, and this one end plate. Now we will discuss each parts in details. Here generally head part, here head part. The shape of the head part is hexagonal or icosahedral in shape. This head part contain genetic material, that is double-stranded DNA, and this head part is made up of with small subunits, and these small subunits are called capsomers. Capsomers are also, we can use another term, cashmer, and there are 2000 capsomers are made this head part. 2000 capsomer or cashmers. In tobacco mosaic virus, there are 230 capsomers are present, but in bacteriophage we have only 2000 capsomers or cashmers. So this is all about the head part here. So this is the head part. Here we have double-stranded DNA as a genetic material, and this head part role is to protect the genetic material. So this head part are capsomes or chiasmus. They helps in protection for the genetic material. Now coming to collar part, and this collar part is a narrow connecting region between head and tail. So it is a collar part is a connecting region between head and tail. Now coming to tail. So the tail part is surrounded by, or it is covered by, a sheath called tail sheath. Tail sheath. And these tail sheath made up with 144 subunits. The subunits also we can use the term capsomes. So there are 144 subunits or capsomes are made the tail sheath, and tail sheath which is present around the tail. And these 144 subunits or capsomes are arranged into 24 annual rings. They are arranged into 24 annual rings. So this is all about the tail sheath, and at the end of the tail sheath, here arranged into 24 annual rings. Annual rings. In each rings there are 6. In each rings have the 6. And at the now coming to last part, that is end plate. End plate are also called base plate. Base plate, and this part is present at the bottom of the tail, which is present at the bottom of the tail. Bottom of the tail we have seen this end plate or base plate, and from which bottom of the tail or base plate there are two structures which arise. They are tail fibers, and second structure, one more structure, that is called tail pins. Tail pins. Here from this region, base plate, like this, 3 pairs of tail fibers or 6 tail fibers which is arise. See, this is 3 pairs or 6 tail fibers, and at the base of the end plate there are only 3 tail pins are present here. 3 tail pins, and here the role of tail fiber is, it helps to attachment. It helps to attachment to the bacteria host cell. Helps to attach bacterium host cells. And the role of tail fiber is, and it secrete one enzyme, viral enzyme. Tail pin only secrete a viral enzyme. The enzyme name is lysozyme, and these lysozyme enzymes are secreted from tail pins, and these lysozyme enzymes helps to lysis the bacterial cell wall. It helps to lysis the bacteria cell wall. So this is all about the structure of bacteriophages. Now coming to, we have different types of bacteriophages, like based on which we classify into different categories, we will discuss. Yes. So classification are types of bacteriophages. Types of bacteriophages. Based on 2 criteria, we classified bacteriophage into different groups. First 2 criteria was classification based on genetic material. Here first 2 classification made are based on genetic material. genetic material. So based on genetic material, the bacteriophages are classified into two separate groups. Like some bacteriophage has the main genome as a DNA, hence DNA phase virus, DNA phase, and second one is RNA phase, RNA bacteriophage. So usually most of the bacteriophage has DNA as their genetic material, and even though in DNA also we have two types of DNA strands, that is they are double stranded DNA and single stranded DNA. So double stranded DNA phages, best examples are T even bacteriophages, T even bacteriophages. So these T even bacteriophages, they contain their genetic material as double stranded DNA. T even bacteriophage means in which we have T2 bacteriophage, T4 bacteriophage, and T6 and T8 bacteriophage, etc. Coming to single stranded DNA bacteriophages, example is very important, lambda phase. Lambda phase are also called coliphase, So this lambda phase, coliphase contain the genetic material as a single stranded DNA. Usually mostly bacteriophage contain DNA as their genetic material, but there are some exceptions. There are some bacteriophages, they also contain RNA as their genetic material. So in which again we have in RNA double stranded RNA and single stranded RNA. Now coming to double stranded RNA phase, bacteriophages, like we have pi 6 phages. Pi 6 phage is the best example for double stranded RNA bacteriophage. And coming to single stranded RNA phages are MS2 phages, MS2 phage, and very important bacteriophage which contain single stranded RNA as a genetic material, that is QB bacteriophage, QB bacteriophage. So this QB bacteriophage and MS2 phase both are contain single stranded RNA as a genetic material, but pi 6 phase contain only double stranded RNA as their genetic material. So like that we classified bacteriophages based on genetic material, in which very important we need to remember double stranded DNA and single stranded DNA phages. Okay, right. Now coming to next classification. Next classification made are based on life cycles. So classification, next classification based on life cycle of bacteriophages or reproductive strategy, based on the reproductive strategy. Based on the reproductive strategy or life cycle, bacteriophages again classified into two groups. So they are, first group is lytic or virulent phase. Lytic are also called virulent phase, and second one is lysogenic or temperate or non-virulent phase, phase. And example for lytic or virulent phases are T1 bacteriophages. The best example for lytic or virulent phase is T1 bacteriophages, like T2, T4, T6, T8, etc. And best example for lysogenic or temperate or non-virulent phase example is lambda phase. Lambda phase are also called coliphase. And do you know this T even bacteriophage and lambda phage both contain their genetic material as a DNA, but here the major difference is T even bacteriophage has double stranded DNA and lambda phage DNA has single stranded DNA. So this is the another classification which is made based on the life cycle or reproductive strategies of bacteriophages. So, this is all about the bacteriophage introduction and structures and their classification. Thank you.